to spend much time during this nutshell video talking about the components of blood, because you can read that in your textbook and it's fairly straightforward what platelets do and how the coagulation cascade works, for example. Later on, we will get into the immune system, which deals with white blood cells. What I want to talk about here is hemoglobin found in the red blood cells, in terms of what the hemoglobin is doing to help transport the gas oxygen in the bloodstream. So this section is really a top physiology section, I would say. And it's something that you need to spend some time getting a foundation with memorizing some numbers. And I hate that, but until you memorize, for example, how hemoglobin behaves, what the points are on the graph for the saturation curve, then if you don't memorize it first, then you can't use it in a physiological application. So let's start by talking about hemoglobin as a protein, which is a, you can find this figure, this diagram pretty much anywhere. It's a big 4 amino acid protein. It has what's called quaternary structure. So that is one hemoglobin, which has 4 separate amino acid chains. And at the center of each one is a heme group that contains iron. And that's the pigmented part. And so for one hemoglobin, that's my abbreviation, HB, one hemoglobin protein, each heme group, and there are 4 of them, can bind to an oxygen molecule. So an oxygen molecule looks like this: O2, 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 O2. That's 4 oxygen molecules for one hemoglobin. And so as hemoglobin travels throughout the body, what you will see is basically this equation of hemoglobin binding to oxygen gas, and it becomes what is called oxyhemoglobin, which we will draw like this, HbO2, because now they are bound together, so that would be HbO2. Because hemoglobin is a big, huge protein, and proteins have specific behavior based on their shape, sometimes hemoglobin is not binding to 4 oxygens. It might be binding to 3, and that would be 75% saturated. Or it might be binding to 2, and 2 would be full and 2 would be empty. That would be 50% saturated. Or hemoglobin protein might be binding to 1 of the 4 oxygens that is, has potential to bind to. That would be 25% saturated. So this equation of hemoglobin binding to oxygen, if it were to be completely saturated, the way I would draw that is I would draw it as a circle representing our hemoglobin protein, and then 1, 2, 3, 4 oxygens. But you need to remember that the key to hemoglobin transporting oxygen is it binds to the oxygen, that would be at the lungs, and then it unbinds from the oxygen, so this must be reversible. There's no enzyme to catalyze this reaction, but indeed hemoglobin will bind to oxygen and will unbind from oxygen, which is what it needs to do in order to release the oxygen to the tissues of the body. So we're going to talk about how the oxygen concentration here, this is the key ingredient, if you will, to determine how much of the hemoglobin is binding. Okay, so we don't say oxygen concentration. What we're going to be saying is partial pressure of oxygen, because out here in the air that we breathe, there's a whole bunch of gases, and we want to pull out the part that is just due to oxygen and look at that. So we're going to be just looking at oxygen in this big mixture of gases. So we would say, what's the concentration of oxygen more correctly as what is the partial pressure of oxygen? So PO2 is the partial pressure of oxygen, which is the same thing as like the concentration. So, for example, if we have the alveoli here, which is where the air that you breathe ends up, we're just going to be looking at oxygen because it's really all that we care about. And we want that oxygen to be able to cross over the pulmonary capillary membranes here and be able to get, so if you don't remember your anatomy, that is the pulmonary capillaries, to be able to get into the bloodstream. Since you already learned that oxygen gas is nonpolar, you know that oxygen moves by simple diffusion and you cannot stop it. So as long as the PO2 here is high compared to the PO2 here, you will have a gradient. And that's like a concentration gradient. Only because we're talking about gases, it's a partial pressure gradient. And so the oxygen will move into the bloodstream by simple diffusion. You need to remember what you learned about Fick's law. Okay, so you need to review that, Fick's law, which says that this rate of diffusion will be faster if you have a stronger gradient. So some people, they breathe oxygen in order to make this really high. If they're about the same, then you have a small gradient, they don't move. You want to make that a lot higher than the bloodstream so it will have a good high rate of diffusion. So Fick's law, you can review that, has to do with the rate of simple diffusion here. Surface area also comes into play in the respiratory system. If you have more surface area, then you have a stronger, higher rate of diffusion. People with emphysema have less surface area, so they have decreased rate of diffusion. If you wedge a layer of water in here, that'd be like pulmonary edema. That would make the distance between these two membranes greater. And if you increase the distance, it decreases the rate of diffusion. So use common sense there, and please review Fick's law. It's in lots of places in your book. Okay, so let's go back to hemoglobin. And I want to look at the behavior of the protein alone, and then we will look at it in the human body. So let me scoot down here for a second, and let's pretend that we're in the laboratory, and we are going to look at, actually, I'm going to use this space later on for something else. So let's go up here to the top. I have a little bit of space up here, and I can use this space up here. I'm trying to get it all on one page, which is maybe not a good idea, but we'll see. Let's pretend that you're in the lab, and you're doing an experiment with this protein, hemoglobin, which is a big, huge protein. And you want to test it to see how it behaves in terms of its binding to oxygen at various concentrations of oxygen or various PO2. So you can do this in the lab. It's been done before. You can look at the data in your book. So I'm going to draw for you what the data looks like, and then you need to memorize it. So your independent variable on the x-axis is going to be the various concentrations of oxygen, which we call the partial pressure of oxygen. And in order to get a really nice, clean curve that looks accurate, you should make your increments very even. So we're going to do these in 10 millimeters of mercury. That's our unit. 10 millimeters of mercury increments. So there would be 10, and then 20, and then 30, and then 40, 50, 60, 70, 80, 90, 100. Okay, and I'm not going to go above 100 because 100 is really the highest PO2 you will see in the human body. It's what you see here in the lungs because that's the place of highest. So you might label some of these. We'll be putting some of these numbers, 40, 50, 60, 70, okay. So those are 10 millimeters mercury increments. Now, what we've done in the lab is we've taken hemoglobin and we've dropped it into this environment of varying PO2s. And then we're looking at the hemoglobin in the lab to see what does it look like, what is its saturation. So over here on this axis, we're going to write hemoglobin saturation, and that is a %. So again, if it has 1 oxygen of 4 binding, that's 25% saturated. If it has 2 of 4, that's 50%. If it has 3 of 4, that is 75%, and 4 of 4 would be 100%. So if this is 10, 20, 30, 40, 50, 60, 70, 80, 90, 100, 100% saturated would look like it has 4 oxygens bound to the hemoglobin. Okay, all 4 out of 4 sites are filled. And 50% would of course be 10, 20, 30, 40, 50. 50% would have 2 out of the 4 binding sites filled with oxygen. And 25%, you could find that between the 20 and the 30 right there. Easier. Okay, so sometimes students like to see a little graph, like a little, not a little graph, a little schematic of what that looks like. So I always draw the hemoglobin over there with 25% looking like 1 oxygen, 52, 75, 3, and 104. Okay, let me see if I can figure out how to make that bigger for you. Maybe I can, and that would be cool. Oh, look at that. Okay, so maybe you could see the curve bigger if I just put it right there. Because what you need to do is you need to memorize these numbers, and then you can use them. So 30 is 60, 60 is 90, 90 is 100. I'm going to say that again in case you've never heard it before. 30 is 60, 60 is 90, 90 is 100. So PO2 of 30 is 60% saturated. Okay, 30 is 60, 60 is 90, and 90 is 100. 60 is 90% saturated, and 90 is 100. Then if you can put those points on your graph, you can use it. And that's the whole goal here, is to memorize how hemoglobin behaves, so then when you see it in the human body, you can predict what's happening based on its behavior. 90 is 100. Okay, and so I would like for you to practice drawing this curve. And I would like for you to find at least those 3 points. 30 is 60% saturated, it's about right there. 60 is 90% saturated, and 90 is 100% saturated. And indeed, the curve is going to be flat like that. Oops, sorry, my pen is not working very well. Flat like that, and then it starts to drop off. And you can look at this better in your book. But it's really important that you can draw this curve. It's just the first step to being able to understand. That's how hemoglobin works. It's a protein. It's very predictable. And of course, this does follow along with the equation over here that we drew. Let me jump out of my zooming here. And it does follow along with the equation, which is that hemoglobin plus oxygen will bind to the hemoglobin. So hemoglobin by itself plus oxygen will bind to the hemoglobin, making oxyhemoglobin. If you increase the PO2, according to the curve over here, you will increase the binding or shift to the right. So the amount of hemoglobin that binds, so I'm going to say the PO2 determines the hemoglobin saturation, which is over here, what you're seeing on the right, okay? And we always give hemoglobin saturation as a percent. Okay, so I'm trying to get rid of this box here. I don't really know how to. Oh, there, it went away. Okay, very good. So the next thing that we need to do is take this information, this data that you learned in the lab over here, and we need to look at the human body, okay? So I'm going to leave the curve up there, and I'm going to come down here and review some anatomy with you, and hopefully it will come right back, and you'll remember that when the blood leaves the pulmonary capillary, it's going back to the heart, and eventually it will end up going to the body with this oxygen. And I'm ignoring the heart for this diagram because it's not important. What you need to know is the blood vessels that take that oxygenated blood to the body, they are systemic arteries. Okay? Systemic arteries take the oxygenated blood to the tissues of the body. And when they get there, there will be another set of capillaries called systemic capillaries. So the systemic capillaries are going to be right through the tissues. So you have these cells right here which need the oxygen. And so again, the oxygen needs to be able to diffuse across these membranes, which is simple diffusion. It easily does that because it's nonpolar, and it crosses through the tails of the phospholipids very easily. So these are tissues which have a variety of cells. Especially if those cells use aerobic respiration, they need the oxygen. And the capillaries will merge into systemic veins, okay? And the systemic veins will take the deoxygenated blood back up to the heart, which then pumps it over to the lungs. And I'm ignoring that right now because it's not important. And it ends up in the pulmonary capillaries again. So this is the cycle that I want you to be able to trace hemoglobin through. Okay, and if the PO2 is high here, oxygen will move from high to low into the bloodstream. Until when? Until it's equal on both sides. That's the definition of diffusion. So you need to know these numbers, that the PO2 is 100 here, millimeters mercury, and the PO2 of the blood as it enters the pulmonary capillary is going to be 40 or lower. 40 or lower. So of course we have 100 versus 40. It's going to go from high to low until it's equal on both sides at 100. So eventually it's going to equilibrate so that both sides will be 100. Okay? And your hemoglobin never leaves the bloodstream. So the hemoglobin that's hanging out here in the bloodstream inside your red blood cells, at that moment when the diffusion happens, what happens to the hemoglobin? Well, you need to look up here at your curve and tell me what happens to hemoglobin at PO2 of 100. It will be 100% saturated. It will be 100% saturated. And this is a good thing. We want the hemoglobin to be 100% saturated as it leaves the lungs. So this is a normal, healthy person, not at altitude, not with COPD, that has 100 millimeters mercury PO2 here, and it's coming in at 40. So high to low movement until it's equal on both sides at 100. And then that will make the hemoglobin behave in such a way that it binds to the oxygen in its environment of the bloodstream right there. And it takes it out of the plasma. And if you didn't have hemoglobin, you wouldn't be able to carry enough oxygen in the plasma alone. You can look at those figures in your book also. Okay, so here we go. We're looking at the hemoglobin in the systemic arteries, and it looks just like that. 100% saturated because it's looking like whatever happened up here. In other words, there's no change. There's no change in PO2. So that's what is governing the behavior of hemoglobin based on this graph. And so we don't see hemoglobin change until we get down here to the tissues. And what we see here is at rest, the PO2 here is going to be 40 millimeters mercury. Okay, 40. And that is going to be something that is a new environment that the hemoglobin is passing through. First of all, think about what you learned about oxygen diffusion. Okay, so we're going to have a PO2 here in the plasma that is looking like 100. So it's going to be the region of high, and PO2 here of 40 is looking like the region of low. Okay, and your hemoglobin is coming in here, and nothing has happened until, so let's draw it the same. Nothing has happened until the PO2 changes. And as this oxygen diffuses down its partial pressure gradient by simple diffusion across the phospholipid bilayers, as that happens, the PO2 over here in the bloodstream is going to decrease because it's moving out until it matches on both sides, and now your PO2 is going to be 40 over here. So it will match 40 and 40. And your hemoglobin is going to say, whoa, I don't saturate oxygen 100% of PO2 of 40. Can you look at the graph up here and tell me what the oxygen saturates at, what the hemoglobin saturates at PO2 of 40? Okay, so here we are in the resting tissues. PO2 of 40, hemoglobin saturation should be 75% saturated. Okay, so come down here and show me that you understand that by saying what happened to the oxygen that fell off. Okay, it got delivered. Okay, it got delivered, and that's how hemoglobin indeed delivers the oxygen. So now it is free, and it can move over here from high to low. Okay, and so as the hemoglobin passes back up, it is going, if you're resting, if your cells are resting, it is going to look like 75% saturated. Whatever the PO2 is here in your cells, that is going to dictate how much saturation your hemoglobin becomes as it passes through here. Okay, because the PO2 determines the hemoglobin saturation. So here's PO2 of 100. That determines the hemoglobin saturation of 100%. If you have COPD or you're at altitude and your PO2 here is not 100, then the hemoglobin saturation will maybe not be 100%. And you can look at those book problems, those application problems I've given you to see what kind of issues could arise there. But whatever happens here with the PO2 is going to dictate how your hemoglobin saturates with oxygen here in the pulmonary capillaries. Then as it moves through systemic arteries, nothing happens to it because there's no PO2 change here. When it gets to the tissues, whatever the tissues are doing will give you their PO2. So if they're resting, the PO2 is 40, but if they're exercising, it will be less than 40 because exercise means using more oxygen. So I'll say will be less than 40. So you can imagine if you're exercising and your PO2 is 30, up here on your graph, what do we see? We see that PO2 of 30 is going to be a saturation of about 50%. And so that's good because it means that the hemoglobin over here will deliver 2 molecules of oxygen. So remember, the hemoglobin doesn't leave the bloodstream. It just keeps going in a circle here, but it is changing its saturation based on the various encounters it has with the PO2 here and the PO2 there. Okay, so I've drawn it for you at rest, and if you wanted to look at this up here, you could say, okay, well, at rest, the hemoglobin is coming in to pick up the oxygen, 75% saturated. So it looks like this at rest, and it only picks up 1 free oxygen. Okay, but if you had exercising tissues, then it was probably delivering more, and so it'd be coming into the pulmonary capillaries less saturated. But this is what it would look like at rest, for example. Okay, so you would have 75% saturated coming in here, and then it's not so dramatic. So actually deoxygenated blood is not completely deoxygenated. That's what the hemoglobin looks like. Okay, other things you need to remind yourself of, this is a protein, and so if you look at the PO2, it behaves in that way. It's all based on shape. The other things we learned earlier this semester together are about pH changes, can also change the shape of a protein. Protein and temperature. Increase temperature changes can also change the shape of the protein. So, for example, if you increase the temperature or you decrease the pH, that will cause the curve to shift to the right. And that's called the Bohr shift. So, and it's just because it's changing the shape of this protein. Oops, I'll say shape changes. Okay, so you need to think about pH and temperature. So that's called the Bohr, named after Bohr shift. Okay, and I always think about it in terms of if you're exercising, you would get a decrease in pH by acid production and an increase in temperature. And both of those are going to cause the hemoglobin to have lower saturation, which means it more freely releases its oxygen when it gets to the tissue. Okay, so even if PO2 stays the same at 40, the hemoglobin saturation is going to be lower for every point on this curve, and that is if you increase the temperature, decrease the pH. Okay, so that would be the Bohr shift to the right for increased temperature, also for decreased pH. Okay, so there's a lot there. Let me know if you have questions, but that is the overview of how hemoglobin behaves in terms of oxygen transport. I hope it was helpful.